Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET entrance exam question paper. This is for the PG entrance, and I will be discussing the subject Life Science, which was conducted in the year 27th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Life Science being discussed for this particular entrance exam, you can go to my channel Omni Bio Entrance, see for the playlist. From the playlist, select CUET and you will find your question papers being answered there. Now coming to the next question. Given below are two statements. One is labeled as assertion and the other is labeled as reason. Telomeres are unique structures at the ends of linear chromosomes. Of course, telomeres are replicated by telomerase, a specialized polymerase that carries its own RNA template. That is also correct. In lagging strand in prokaryotes, first an RNA primer is synthesized, by primase. Like leading strand synthesis, DNA polymerase 3 binds to RNA primer and adds deoxyribonucleotides. That statement is also correct. But in assertion, they have mentioned about telomeres, and in reason, they have mentioned about DNA replication in lagging strand. They both are not related, but both the statements are correct. And the reason is not explaining the assertion. So both A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then next one. Given below are two statements. One is labeled assertion, reason. Assertion. In preparatory phase of glycolysis, phosphorylation of glucose takes place and converts into glyceraldehyde 3-phosphate. Of course. Hexokinase, like many other kinases, require Mg2+ for their activity. That's also correct. In the payoff phase of glycolysis, under aerobic condition, 2 molecules of NADH are formed in cytosol along with 2 molecules of pyruvate and 2 molecules of ATP. That is also correct, but the reason doesn't explain the assertion. Both the statements are correct. So A and R are correct, but R is not the correct explanation of A. Answer is option 2. Now before coming to the next question, I will tell you what is site-directed mutagenesis. So site-directed mutagenesis, it is used to introduce specific targeted genetic mutations into a DNA sequence. Now you have got a DNA sequence. You want to mutate it, you go for site-directed mutagenesis. Now first what you do is, you design, that is choose specific site and mutation. So this is what you have to mutate. Now you choose the mutated region, the region which has to be mutated. Then you design oligonucleotide primers complementary to the target sequence, incorporating the desired mutation in the primer. That is like this, mutagenic primers are being designed. Then you amplify the target DNA using mutagenic primers. So this is amplification process. You amplify the target DNA. Then you clone them into the then you insert the mutated DNA into the desired plasmid or a vector. Then you confirm the mutation by sequencing. This is what happens in site-directed mutagenesis. Now one more thing I want to tell you is that here the DNA sequence is mutated. So naturally the protein that is being produced from this particular DNA sequence will also be mutated because your amino acid sequence is mutated. Okay. That is site-directed mutagenesis. Now coming to the question. Given below are two statements, assertion and reason. Specific amino acids may be replaced individually by site-directed mutagenesis. I already told you that. If you want to remove one particular amino acid, you can go for side-directed mutagenesis. This powerful approach to studying protein structure and function changes of amino acid sequence of a protein by altering the DNA sequence of a cloned gene. That is what I explained just now. Through side-directed mutagenesis, you mutate one particular base of the DNA. It will result in a change in the amino acid sequence, which will result in the change in the protein, and it will result in the change in the structure of the protein as well as function of the protein. That is what they have mentioned in assertion. Then next one. When suitably located restriction site A are not present in the sequence to be altered, an approach called oligonucleotide directed mutagenesis can create specific DNA sequence change. Now, oligonucleotide directed mutagenesis, it is a used to technique that is used to introduce specific DNA sequence change when suitable restriction sites are not available. Now here also you design an oligonucleotide primer with desired mutation. You synthesize that primer after designing the primer. Then you anneal the primer to the target DNA. Then you extend the primer by using DNA polymerase. Then you transform the cells with the mutated DNA. Then you isolate DNA from the transformed cells and you confirm the mutation via sequencing. So here both A and R are true and R is the correct explanation of A. Now the reason it explains assertion A. So the answer here is option 1. Then next one. Assertion. The thylakoid reaction of photosynthesis take place in the specialized internal membranes of chloroplast or thylakoids. Yes. The end product of these thylakoid reactions are used for the synthesis of sugar in the carbon fixation reaction. That is also correct and it explains the assertion. So both A and R are true and R is the correct explanation of A. Then next one. In a healthy body, RBCs can live for several months or years. Assertion itself is false. RBCs live for approximately 120 days, not for years. So assertion is wrong. A is false. RBCs are responsible for transportation of oxygen and some amount of carbon dioxide also. That is correct. So A is false, R is true. Your answer is option 4. Then next one. A hormone is a molecule that is released in one part of the body but regulates the activity of the cells in other parts of the body. That's correct. The circulating body blood delivers hormones to cells throughout the body. That is also correct. So, and it is the reason for A. So both A and R are correct and R is the correct explanation of A. Answer is option 1. Then next one. Phytochrome is most abundant in etiolated seedlings. Correct. Most biochemical studies have been carried out on phytochrome purified from non-green tissues. Non-green tissues means etiolated. So A and R are correct. R is the correct explanation of A. Answer is option 1. Now identify the correct sequence of genome of different organism in increasing order. Homo sapien, that should come in the end. So A has to come in the end. So your option 1 is gone. We will see the others. So first one will be Escherichia coli. That's correct. Then comes Saccharomyces cerevisiae D. Then Caenorhabditis elegans E. Drosophila melanogaster and finally Homo sapiens. So it is answer is option 3. Then identify the correct sequence of protein components on mitochondrial electron transfer chain. So out of these first one will be NADH dehydrogenase. So 1 and 2 options can be ignored. C. Okay, C. NADH. Then comes succinate dehydrogenase. So 3 also gone. 4 is the correct answer. A. Succinate dehydrogenase. Then comes ubiquinone cytochrome C oxidoreductase. And finally cytochrome oxidase. Answer is option 4. Then correct sequence of the southern blotting procedure as applied to DNA fingerprinting is. So first what you have to do is you have to digest the DNA with specific restriction endonuclease. Then you have to run an agarose gel. Then you have to transfer it to a nylon membrane after denaturation. Then you have to incubate the nylon membrane with a radio-labeled probe and finally expose it to X-ray film for result. So here first one will be E. So 3 and 4 options can be ignored. E will be digestion of chromosomal DNA. That's correct. Then after digestion you have to separate the DNA fragments by agarose gel electrophoresis. B. So EB, your answer is option 2. Then comes C, that is denaturation of separated DNA and transfer to nylon membrane. Then incubation of the nylon membrane with radio labeled DNA probe and exposure of the X-ray film to membrane for analysis of results. So E, B, C, A, D. Answer is option 2. Then next one. Correct sequence of intermediates formed in Calvin cycle is 1,3-bis-phosphoglycerate, 3-phosphoglycerate, glyceraldehyde, 3-phosphate, ribulose 1,5-bis-phosphate, dihydroxyacetone phosphate. So the correct sequence you have to see in Calvin cycle. So if you see option 2, ribulose 1,5-biphosphate will be the first one. Then comes 3-phosphoglycerate. Then comes 1,3-bis-phosphoglycerate. Then glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So answer here is option 2. If you learn the pathway, you can answer these questions. Then correct sequence of vertebrae arranged in a vertebral column in the human is. So the first one will be atlas vertebrae, E. Then comes axis vertebrae, then thoracic vertebrae, then lumbar vertebrae and sacral vertebrae. Answer is option 1. To obtain large number of copies of DNA through PCR includes the following steps. So what are the steps involved in PCR? First, it should be double-stranded DNA with known ends, A. So everywhere A is given. Next will be everywhere B is given. So separate chains by heating at 95 degrees Celsius. So B is also correct. Then what you have to do? Then you have to cool to allow primers to form hydrogen bonds with ends of the target sequence. That is annealing. First is denaturation, then comes annealing. That is E. After E, then you have to add extension by Taq polymerase. Denaturation, annealing, then comes extension by Taq polymerase. So addition of heat tolerant Taq polymerase and nucleotides and heat to 72 degrees Celsius. So C. Then comes addition of nucleotides to primers using single strand as template. D. So answer here is option 3. Then next one, the lytic cycle of phage T4 has. So first one will be that phage has to infect the bacterial cell, then it has to give out its DNA into the bacterial cell, then the DNA of the phage, it will integrate with the DNA of the bacterial chromosome and direct the bacterial chromosome to produce enzymes which are necessary for the phage DNA or phage replication. Then it will lyse the bacterial cell and come out. That is what happens. Okay, we will see what are the options. So first one, T4 makes attachment with specific surface protein through tail fibers. B. Oh, everywhere B is given. Okay, B. Then D. Contraction of tail sheath. DNA enters into the cell leaving capsid outside. So B, D. Then phage directs production of phage protein and copies of phage genome with the help of host and viral enzymes. E. Then self-assemble of the three different proteins to form a phage component. A. Then production of enzyme to damage bacterial cell wall so that it can lyse the bacterial cell and come out. So answer here is option 3, that is B, D, E, A, C.